those are your objectives. I'm not going to read through all of those because we're going to be talking about them. So the nervous system, like everything in the body, when scientists find a thing that's complicated, what we do is we find a way to divide it up, right? So that we don't have to think about the whole complicated thing all at once. So in the nervous system, we break it up a variety of different ways. So central versus peripheral is certainly a valid anatomic way of dividing up the nervous system, right? Well, then we have, under peripheral, we have our somatic nervous system. That's the one we think about most of the time. That's the nerves that you're identifying in the anatomy lab. Carrying voluntary muscle control and bringing in conscious sensory information into the brain. Now, the autonomic nervous system, which is what we're going to talk about today, is technically part of the peripheral nervous system because it exists outside of the brain and spinal cord, okay? And its job is really homeostasis. The autonomic nervous system works in command and control. Our bodies are a complex and changing set of variables in an environment that is also complex and changing. So it means that there needs to be intelligent control between the different areas of the body. And it's the autonomic nervous system that primarily manages this aspect of staying alive as a human being. So underneath autonomic nervous system, we have two branches, the sympathetic nervous system, or SNS, and the parasympathetic nervous system, or PSNS, okay? Sympathetic nervous system tends to be an activator of body activity. Parasympathetic nervous system tends to be a rest and repair sort of signaling, okay? And we'll talk more about that as we go. Now, other ways that we break up the nervous system, just to be complete, we have central versus peripheral. We have afferent versus efferent. In other words, what's coming in versus what's going out. And then we also can talk about it in terms of sensory versus motor, right? But more on that when we get into the nervous system. Okay, so if we look at a simplified look of the somatic nervous system, we can see that we have signals that come from the brain, have a synapse either in the brain stem or in the spinal cord, and then a second neuron, a lower motor neuron, then goes out to the skeletal muscle to activate that. That's what we were just talking about, right? The lower motor neuron, it ends at the neuromuscular junction. Okay, so it connects with what we were just talking about. So we have one synapse between brain and muscle, or brain and effector. Okay, so a muscle is a kind of effector. It does something. An effector is just something that does something. Okay, so if we look next door at the autonomic nervous system, we see that we gain a synapse. So instead of having one synapse between brain and effector, we have two synapses between brain and effector. So here, information or signaling from the brain, again, goes to the brain stem or the spinal cord. And then our second stop is at what is called a ganglion, an autonomic ganglia. Okay? Ganglia is the plural. Ganglion is the singular, all right? So an autonomic ganglion is one thing. At the autonomic ganglion, what we have is we have the ability for the body to bring multiple signals together before sending a final signal out to the effector. So, because a ganglia, the definition of a ganglia, or a ganglion, is it's a collection of neurons found outside the central nervous system. Okay, that should sound familiar. You should have heard that in your undergraduate careers. Where the ganglia are is going to be dependent upon whether they're part of the sympathetic nervous system or the parasympathetic nervous system. So more on that here in a minute. Okay? So because of this extra synapse between effector and brain, we have to have an additional bit of vocabulary. Okay, so we have the preganglionic neuron comes from the CNS to the ganglia. The postganglionic or ganglionic, those are synonyms, neuron or fiber comes from the ganglia and goes to the effector. Okay? Now, typically in the autonomic nervous system, the postganglionic neurons are not myelinated. Remember, the autonomic nervous system is ancient, old, evolutionarily old, okay? Reptiles, fish have autonomic nervous systems, okay? So this is very old system. So we don't have any myelin. That should tell you immediately that this ain't a fast system, okay? It's going to take seconds to minutes to have an effect, as opposed to the somatic nervous system, which can, of course, have effects in milliseconds, right? Okay, so autonomic nervous system, two primary divisions, and a third one which people have started talking about differently in the last 10, 15 years, all right? Sympathetic, parasympathetic, we talked about already. Enteric, the nervous system, research has shown, the nervous system has as many neurons as the spinal cord does. The GI system, did I say nervous system? Okay. The gastrointestinal system has as many neurons in it as the spinal cord does. So, and that makes sense because, the GI tract is this great big long tube, muscles, glands, a lot of stuff goes on to pull nutrients out of food, all right? So, the enteric nervous system is really the third division of the autonomic, and it controls the gut. We won't talk much about the enteric system in this section. We will when we get to the gastrointestinal system. And I've got kind of a long list here of what this autonomic nervous system does, but the bottom line is it brings in sensory data and it puts out regulatory control, right? To control things like blood pressure, heart rate, breathing, our nutrient and gas concentrations. All of those things are touched upon by the autonomic nervous system. Now, the ANS is involuntary. At least, by definition, it is. In other words, in the normal case, you can't change your heart rate by thinking about it. Now, the reason I'm being a little hesitant on this is you can actually learn to, right? There are many examples of people who've spent a long time learning to do things. Like I just read about a guy who can change his pupil size consciously. So you can learn to do this stuff, but that isn't the normal way that this works. It's usually involuntary. So all viscera have both sympathetic and parasympathetic innervation. These systems, it's not like one system works on one thing and the other works on the other. They both work on everything. So what we see is what's happening in a particular organ or what's happening in the body is the balance of parasympathetic effects and sympathetic effects. They're both happening at the same time. Now, one usually predominates over the other, right? Because the body tries not to fight itself. So we don't really want full sympathetic and parasympathetic activation at the same time. So as one goes up, the other goes down. But both are always present. So for example, if I switched off your parasympathetic nervous system, your heart rate would go up because your parasympathetic system is always pushing it down. Do you see? So if I turn it off, it'll pop up. And more on that as we go into the specific organ systems. I spend a little more time on the autonomic nervous system than I do on other topics in the nervous system because of all the nervous system talks, this has the most relevance to your future life as prescribing providers, okay? There are just oodles of medications that have direct effects on the autonomic nervous system, like that's what they were engineered to do. So for example, many of you will have heard of a beta blocker, right? A beta blocker is a medication that is used to treat hypertension. It's also used to treat post-myocardial infarction. It has a role in diabetes, lots of things. Okay, what is a beta? Well, it's a beta receptor. The beta receptor is part of the sympathetic nervous system. Do you see what I mean? So here is a very commonly prescribed medicine whose effects are directly related to how the autonomic nervous system works. So that's why I slow down and talk about this a little bit right here at the beginning of the course. So cardiovascular for sure, pulmonary pharmacology. Albuterol is a medicine that we use in asthma. Albuterol is a beta-2 agonist. So once again, it's directly affecting a receptor that's part of this autonomic nervous system. Now, this is not pharmacology. Okay, the only reason that I'm going to make these clinical connections is so that you can see why it's relevant. I'm not going to be asking you about medications, not yet anyway. That'll be in the spring and the summer. Okay, so from the sort of top down. So we have our three divisions, sympathetic, parasympathetic, and enteric, right? Sympathetic, also known as the fight or flight system, right? Because it's stimulatory. Also known as the thoracolumbar division because of where it emerges from the spinal cord in the thoracic and lumbar region. The parasympathetic, also called the rest and digest system, or the craniosacral division. Cranio top, sacral bottom, right? Cranium, sacrum, that's where those nerves emerge. So anatomic names and functional names, as well as the standard sympathetic and parasympathetic. And then the enteric system is made up of all those neurons that control the GI tract. And interestingly, the GI tract works almost completely independently of the central nervous system. And frankly, that's probably a really good thing because our CNS has plenty to do as it is. So the sympathetic and parasympathetic divisions both have influence on the enteric division, right? But neither control it. Okay, so for example, the sympathetic nervous system tends to turn the enteric system down. The parasympathetic nervous system tends to turn it up, right? Which is why we call parasympathetic rest and digest. Okay, so that vocabulary. We've got our preganglionic neuron here. This is in the CNS, right? So spinal cord typically. We have our preganglionic fiber. This is the axon of the preganglionic neuron. It's going to go out to the ganglia. The ganglion, one is ganglion, okay? And then from the ganglion, we have a fiber that goes out then to the effector organ. That fiber that goes out to the effector, it's called postganglionic fiber or ganglionic fiber. It depends on whether you think of the axon as part of the cell that's in the ganglion or not, okay? Either one is fine. I tend to call it the postganglionic fiber because it makes it very clear that we're after the ganglia, okay? And then the target effector, what's going to be changed by this system, is then in or near the organ of interest, okay, which we have some pictures of here in a minute. All right. So here's the sympathetic division. Thoracolumbar, so we're in the middle of the spinal cord, right? Where are the ganglia? Well, we have the sympathetic chain, which you will see in the anatomy lab in some weeks. We have the collateral ganglia, which are in the abdomen and pelvis. And then we have the adrenal gland, okay? Now, target organs we're going to find all over the body. So one of the patterns that we see, all of these ganglia are relatively close to the spinal cord. So we get our first pattern. The sympathetic nervous system has short preganglionic fibers and long postganglionic fibers. All right? The parasympathetic division is going to be opposite of that. Okay? Now, we also have the sort of special case of the adrenal medulla. The easiest way to think of the adrenal gland is the adrenal gland is a ganglion that has, instead of using axons to transmit its information, uses hormones instead. Okay, so that's epi and norepi, and we'll talk about those here in a little bit. So sympathetic division, short preganglionic, long postganglionic. Now, parasympathetic, okay, so craniosacral area, you'll notice the whole middle is missing. That's thoracolumbar, that's sympathetic. Craniosacral, where are the ganglia here? The ganglia of the parasympathetic division are found in or very close to the organs they're going to affect. All right? So, like the ciliary ganglion is actually in the eye, right? The pterygopalatine and submandibular ganglia are in the skull, very close to the nasal glands, tear glands, salivary glands. The otic ganglion is very close to the parotid gland. The intramural ganglia are actually inside the organs that the parasympathetic nervous system innervates. Okay? So if we look at all that, what we see is our second pattern. We have long, in the parasympathetic division, we have long preganglionic fibers. So from CNS to ganglion is a long distance. Very short postganglionic fibers, because the ganglia are pretty much in the organs where they're going to affect. So their output neurons, their output axons are very short. Okay, so long preganglionic, short postganglionic. Same information, different presentation. Okay, so this is sympathetic again, thoracolumbar. Here's the sympathetic chain, okay, which lives literally right next to the spinal cord. It lives on the sides of the vertebral bodies, okay. And then, so we have short preganglionic fibers in red, and then long postganglionic fibers in black. Okay, so this is more of an anatomic representation of the same thing. The other thing I want you to point or to notice is pretty much every organ system is represented here, right? The sympathetic nervous system has the broadest innervation. And some of that is because every single one of your arteries in every nook and cranny of your body has sympathetic nervous system innervation. So that means there are many, many, many more sympathetic neurons than there are parasympathetic neurons, all right? And they're more widely distributed. Same kind of picture. This is parasympathetic, right? So craniosacral, long, very long, preganglionic fibers in red, teeny tiny little postganglionic fibers in black, right? Because these preganglionic fibers are going to go all the way out to their organs of interest, have a synapse and a ganglia that's in or near the wall, and then have very short postganglionic fibers. You'll also notice, if you sort of follow some of these tracks, that this particular nerve, the vagus nerve, cranial nerve 10, contains most of the parasympathetic innervation for all of the organs in the thorax, right? So it's a nerve you're going to find over and over again in the anatomy lab. Vagus means wanderer, okay, that's what that word means. It has the same root term as vagrant, right? So it's called that because it's like everywhere you look in the body you find little bits of the vagus nerve, at least in the thorax you do. Now, up in the head, that's not the case. You know, up in the head, eye, lacrimal glands, salivary glands have their own innervations through cranial nerves 3, 7, and 9. Okay? And then when we get down into the sacral region, we're into the pelvic organs, urinary bladder, reproductive organs primarily. Okay? So, long preganglionic, short postganglionic. The sympathetic nervous system and the parasympathetic nervous system each have their own unique classes of receptors. All right. Now, to remember, a receptor is essentially a chemically activated protein That might be a channel, like we've been talking about with action potentials, like a chemically gated sodium channel, like we see in the at the neuromuscular junction with acetylcholine. Or we can have receptors like these, which affect intracellular function. So ultimately, what a protein receptor has to do is it has to bind to a very specific molecule, because remember, passing through the extracellular fluid and passing through the bloodstream are lots and lots of different kinds of molecules, right? So a receptor has to be specific in order to do its job. It has to be affected by only one or at least a very small number of molecules. That gives the body control over what cells do. Ultimately, the brain needs to affect what a distant cell is up to, right? So the receptor system is the primary way that that's done in the autonomic nervous system. Okay, so we have in the sympathetic system, we have alpha and beta receptors, okay? Inside those big classifications, we have two alpha receptors and 3 beta receptors. An alpha 1 and an alpha 2, and a beta 1, 2, and 3. Okay, so that's a lot of specific numbers and things to remember. So some things to help. Alpha, or any receptor that has a 1 after it, so alpha 1 or beta 1, those are stimulatory receptors, okay? The 2s, alpha 2 and beta 2, are both inhibitory receptors, all right? And then we have beta 3, which is the most recently described of the sympathetic nervous system receptors, and it's only found in one kind of tissue, at least as far as we know, and that's adipose tissue, okay? So that's a little sort of mnemonic or helpful. All right, so let's talk about the alpha receptors first. Now you'll notice in our receptor picture here, we have 2 slots for molecules. It's because the sympathetic receptors can be activated by either epinephrine or norepinephrine. Okay, now you can see by their names that they must be related, and they are. The norepinephrine is really a modified epinephrine molecule. The difference between the two is norepinephrine tends to have a shorter lifespan in the body than epinephrine does. Okay, so norepinephrine is broken down more quickly, therefore its effects have a shorter duration. Epinephrine, on the other hand, withstands some of the enzyme attacks and therefore has a longer term of action. That's why in our resuscitation environment, like when we're trying to restore cardiovascular and pulmonary function in a patient who's had like an MI, we reach for epinephrine because epinephrine has a longer time of action than norepi does. Okay, and more on epi when we get to the end here. Okay, so alpha receptor affected by norepi or epi. The alpha 1 receptor works through a second messenger system. Okay, a second messenger is simply this. A receptor creates a new molecule inside the cell. That new molecule inside the cell then goes on and makes something different happen. Okay, that's what I mean by a second messenger system. So in the case of the alpha 1 receptor, when norepi binds, the alpha receptor creates this second messenger. That second messenger then triggers the release of calcium from the endoplasmic reticulum. Oh, calcium just got released, so we know something's going to happen, right? In this case, we get smooth muscle contraction or gland cell secretion. So to give you a really concrete example, we have lots of alpha 1 receptors on our arteries. Okay, so when the sympathetic nervous system stimulates alpha 1 receptors on the arteries, smooth muscle contracts. Do you think that increases or decreases blood pressure? Increases, right? Because we're increasing the resistance to blood flow. So the alpha-1 receptors help to stabilize our blood pressure by causing arterial constriction. So it's an example of how this system works. The alpha-2 receptor, which I said is inhibitory, it reduces cyclic AMP levels. Remember from biochem, cyclic AMP is a class of second messenger, right? So if we turn down the amount of cyclic AMP that's present in the cell, that results in the cell doing less of what that cell does. So to give you another concrete example, we have alpha-2 receptors in the blood vessels that supply our muscles, okay? So in a fight or flight scenario, I like to talk about a leopard jumping out of a tree right in front of you, right? Woo, suddenly you are the prey, okay? Well, what's going to happen? Well, sympathetic nervous system is going to fire way up. So the alpha-1 receptors are going to be activated to elevate your blood pressure. Okay, that makes sense. So what are the alpha-2 receptors up to? Well, they're going to dilate blood vessels going to your muscles so that the muscles you're going to use to fight or flee, I recommend flee, right, from a leopard, are going to have reduced restriction and therefore increased blood flow to supply those muscles that you're going to fight or flee with oxygen and fuel to burn, right? Okay, so those alpha-1, alpha-2 are found, alpha-1s are the most common of the receptors found in the body. They're found in more places, they're found in most tissues, they're sort of the standard. Alpha-2 receptors are found in less places, and the places they're found are usually related to needing to respond to a fight or flight scenario. So we have inhibition. Now, if we flip over to the beta receptors, okay, we have beta 1s and beta 2s. You have one heart, right? So you have the beta 1 receptors are found on the heart. You have 2 lungs, so beta 2 receptors are found in the lungs. Okay, that's a little mnemonic there as well. All of the beta receptors work through the cyclic AMP pathway. Okay, so epinephrine or norepi binds to the beta receptor. Cyclic AMP is produced from ATP by the enzyme called adenylate cyclase or adenylyl cyclase. The beta 1 receptor, if that's the receptor that's found on the membrane, we're going to have an increase in cardiac muscle stimulation. More specifically, we're going to see an increase in heart rate as well as an increase in heart contractility, or how hard the heart squeezes. Okay, and we'll look specifically at how beta 1 receptors work when we get into the cardiac system. Beta 2 receptors found in the lungs, okay, they cause relaxation in the respiratory passages. This is why beta 2 receptors are targeted for asthma rescue medicines, right? Because they stimulate beta 2 receptors. Beta 2 receptors cause the dilation of respiratory airways. Beta 2 receptors also cause dilation of blood vessels in skeletal muscle and the lungs as well. So more on that later also. Then enter the new guy on the block, the beta 3 receptors. Beta 3 receptors are found in adipose sites, in adipose tissue. And when they're activated, they cause the release of fatty acids. Now, what does that have to do with the fight or flight scenario? Fatty acids are the preferred fuel for muscle cells, right? So activation of the sympathetic nervous system causes a release of fatty acids so that the muscles have an adequate supply of fuel to do whatever fighting or fleeing you're going to need to do, okay? So alpha 1, alpha 2, beta 1, 2, 3. On the parasympathetic side of the autonomic nervous system, we only have 2 receptors. We have nicotinic receptors and muscarinic receptors. They get their name from molecules that activate them. Okay, so not surprisingly, nicotinic receptors are activated by nicotine, right? Nicotine found in many tobacco products. Muscarinic receptors are activated by muscarine, much less available than nicotine is, but it is a class of molecules that is actually made by fungi and results in stimulation of the parasympathetic nervous system. The nicotinic receptor is very easy to understand because we've seen a receptor just like it already. So the nicotinic receptor is an acetylcholine chemically gated sodium channel. All right, so just very similar to what we see at the motor end plate in the muscular system. Now, I don't want you to think it is the same receptor, because it's not. The one that we find in muscle is a different protein, but it works in the same way. When acetylcholine binds to it, the receptor opens, allowing sodium to pass through, read membrane potential change, right? So we can make things happen with that. The muscarinic receptor is a G protein linked membrane protein. So here, acetylcholine binding to the muscarinic receptor causes the activation of this G protein, which sits just inside the membrane. That G protein then goes on to activate or inactivate specific enzymes which cause the effect. Okay, so there are lots of different effects of muscarinic receptors. The receptor always does the same thing, which is to activate the G protein. How that receptor's activation affects the cell is going to depend on which cell we're talking about, right? So some cells have one kind of response to activation of G protein. Other cells have a different response, right? So the nicotinic receptors are found at the ganglia of both sympathetic and parasympathetic nervous systems, all right? So if I were to ask you a question like, in the sympathetic nervous system, are there any synapses that use acetylcholine? The answer would be yes, right? Because all of the ganglia for both sympathetic and parasympathetic have acetylcholine as their neurotransmitter. So even though we think of nicotinic receptors as being part of the parasympathetic nervous system, they are also found on the ganglia of the sympathetic nervous system as well. Okay? Now, the muscarinic receptors are found on the effector organs or glands. So whatever the parasympathetic nervous system is going to make happen, that's where we see the muscarinic receptors at the other end. Okay. So, sympathetic nervous system, we covered a lot of this last time, so I'm going to just go quickly. Thoracolumbar, fight or flight, active during periods of stress, arousal, exercise, competition, danger, trauma, anger, fear, right? All situations that are going to stimulate the body into activity. Now, do note that the brain is required to identify these periods of increased stress, right? So if you think about my leopard jumping out of the tree analogy, you got to have a working visual system and a working frontal lobe to make the connection that you are in danger. Do you see what I mean? So even though the autonomic nervous system is entirely subconscious, it can't function without the conscious mind identifying potential risk. Now, it doesn't always work like we want. You've got tests coming up in a few weeks, and during those periods of time, your body is going to act like you're looking at a leopard. Okay? Now, why is that? Because you've identified that test as a stressor, and your autonomic nervous system is treating it like it's a leopard, right? So, the way we get past that, of course, practice, be prepared, right? All that good stuff. But you still have to have the upper level neurons working in order to have this system happen. Now, what do we get out of that? We get increased alertness. That one's very easy to see. You know, if you're suddenly startled late at night when you're getting ready for bed, it's much harder to like just lie down and go to sleep, right? So the sympathetic nervous system gives us a burst of alertness. That's a central nervous system effect. It's not like that's happening because your blood pressure and heart rate are up. It's the other way around. Your blood pressure and heart rate are up because your whole system has been alerted, right? You know, let's say you see a nice big juicy spider, right before you're going to go to bed. All right, so increased alertness, we increase our metabolic rate, heart rate, blood pressure, we increase our airflow. That includes both increased respiratory rate and depth and a reduction in airway resistance. Okay, so we're able to move more air. We increase our sweat production. This isn't always convenient, right? Because nobody likes to be all sweaty. However, what the body's getting you ready for is to fight the leopard. When you're fighting the leopard, you're going to generate a lot of heat. Therefore, you're going to have to get rid of that heat with excess sweat. And then your blood glucose goes up for a similar reason as what the beta-3 receptors do. It's providing fuel to the brain and fuel to the cells of the body to deal with whatever the stressor is. The sympathetic division directly causes a reduction in fear and a reduction in sensitivity to not only pain but other stimuli as well. So a way that we see this is in the phenomena we humans call anger, right? When we're angry about something, we tend to be less afraid and we pay less attention to the details of things, right? Now, this is good in the sense that it helps us to solve whatever the problem is, okay? And the sympathetic division, when it's activated, it reduces our longer-term functions like digestion and the production of urine. The idea here being it's not the time for those things, right? We're going to shift that energy usage into the fight or flee muscles and brain and organs so that we can deal with our friend the leopard. All right, so what about the parasympathetic division? The rest and digest, craniosacral, talked about that already. Most parasympathetic stimuli comes through the vagus nerve, cranial nerve 10. Full stop, right? That is absolutely true, almost all. Other places, cranial nerves 3, 7, and 9 also contain sympathetic nervous system stimulation, and the sacral nerves as well. Okay, the sacral nerves, their primary job is focused in the pelvis. So you've got sexual function, you have urinary function, and then rectal function, right? Defecation control. Parasympathetic decreases your metabolic rate, heart rate, blood pressure, respiratory rate. Easiest way to see maximum parasympathetic activation, look at somebody who's asleep, right? Their digestive tract tends to be quite busy. They're in the digestion mode, but everything else is kind of pushed down so that their heart rate is lower, blood pressure lower, and all that. We get increased blood flow to the GI tract, increased secretion there as well. We get increased motility, and the parasympathetic nervous system tends to stimulate urination and defecation, right? So it's the time of parasympathetic tends to be highest when we feel safe and secure, right? And the basic tasks are done. Now, the parasympathetic also triggers storage of nutrients. So while we're resting and digesting, we're taking the nutrients from that digestion and we're forming glycogen and lipid under the parasympathetic nervous system's stimulation. Okay, so let's look at the comparison of the two. Sympathetic on this side, parasympathetic on this side. First off, little anatomy, right? We've got short preganglionic fibers in the sympathetic, long postganglionic fibers, opposite in the parasympathetic, right? If we look at our neurotransmitters, the neurotransmitters are reflected by the arrows at the synapses here. We can see acetylcholine at the ganglia of both, like we just talked about a little bit ago, right? At the end effectors, though, we see acetylcholine in the parasympathetic system and we see epi and norepi primarily in the sympathetic system. Now, notice down here where it says the neurotransmitters and sympathetic. Normally, it's norepinephrine. We think of the parasympathetic system as adrenergic, in other words, releasing norepi. There are exceptions, okay? Some of the postganglionic receptors, in other words, the end organ effectors, use acetylcholine. Sweat glands, for example, use acetylcholine. And the others use NO. That's nitric oxide. Okay, that's a nitrogen and an oxygen. Nitric oxide we're learning more about all the time. I suspect you'll hear a little bit about it in immunology, maybe even in MedPath as well. But it also can be a neurotransmitter in the sympathetic nervous system. In the parasympathetic nervous system, it's exclusively acetylcholine. Okay, so you don't have the pesky exceptions. And then we've already talked about what those things do. All right. Location of the ganglia. So the sympathetic nervous system ganglia primarily sit right next to the vertebral column in the sympathetic chain. There are some other collateral ganglia, okay? And then the parasympathetic nervous system ganglia are typically found in or very near their target organs, what they're going to affect at the other end. We have our receptor types, right? Alpha 1, alpha 2, beta 1, beta 2. Beta 3 is not on here just because this is a little bit of an older table. But alpha 1, where do we find them? Lots and lots of places. They're very widespread, found in most tissues. They are stimulatory and cause the intracellular release of calcium, which should tell you they're stimulatory, right? Alpha 2s we have in several key places, right? And they're inhibitory through the cyclic AMP system. Beta 1s we find on the heart, the kidneys, the liver, and adipose tissue, primarily the heart. They cause stimulation through enzyme activation, right, through that G protein system. And then beta 2s we find in smooth muscle vessels of the heart and skeletal muscle, and then in the smooth muscle layers of the bronchi and in the gastrointestinal tract, right? So beta 2s are inhibitory. Okay. The nicotinic, all autonomic synapses at the ganglia are nicotinic, okay? And then in the muscarinic, that's only the end effectors. So this is all, whenever I put something like this, you should know these, okay? Because they'll end up being on your exam. Okay. And this one, I was looking at this just a little bit ago, is almost exactly the same as the other table I have in here. So we can effectively skip that. Okay, so the sympathetic and parasympathetic divisions are in a kind of competitive balance all the time. It's not like one's off and the other's on. They're both on, and which one is predominating varies according to what's happening. Okay, so whenever we encounter a graph like this, what I try to do is first tell you what the axes are, okay, so that you can recognize what we're looking at, and then try to walk you through what the graph is showing, at least as far as what I think is important about it. Okay, so on the y-axis is heart rate, right? On our x-axis is time. So we're looking at the change in heart rate over time. Okay, so here at rest, like you all are right now, both of your autonomic nervous system branches are at work. Right? The sympathetic is maintaining sympathetic tone. The parasympathetic is maintaining its tone. So what we see as the heart rate right now is the balance of those two forces. So in our example here, it's 72. All right, well, let's say I switch off your parasympathetic system. Well, we are going to see, let me say instead, let's increase your parasympathetic system just to match my graph. Okay, so we're going to bloop, bloop, bloop. We're going to increase your parasympathetic. What's that going to see? Well, how are we going to see that in your heart rate? We haven't touched the sympathetic, but we've turned the parasympathetic up. So we'll see the heart rate go down, right? Because we've changed the balance. we've pushed the balance in favor of the parasympathetic. Okay, so now let's say we turn the parasympathetic system off. Well, we will see the heart rate go up, right? Because the sympathetic nervous system has less it's pushing against. Well, how do we get our maximum heart rate? Well, the maximum heart rate is going to come when we turn the sympathetic all the way up and the parasympathetic all the way down, right? Then we have very little inhibition and a lot of stimulation, so we get our maximum heart rate, right? So the things that are affected by the sympathetic and parasympathetic, particularly heart and lungs, both divisions are in play, so a change in either division results in a change in the product, right? The outcome, whether it's heart rate, breathing rate, minute volume, whatever. So we have this balance that's in opposition. Okay, so if these systems can change, well, in order for our nervous system to function, we have to have inputs from the world, right? Processing in the central nervous system, and then outputs into our body. So those are put together in what we call visceral reflexes. And we have two different patterns. We have short arc reflexes and we have long arc reflexes. In a short arc reflex, we get to finally see why we have ganglia in the autonomic nervous system in the first place, right? Why bother with the ganglia? Because it allows this to happen. Receptors in a peripheral tissue send information to the ganglia. The ganglia does a tiny bit of information processing and determines whether a response should happen or not, right? So if a response should happen, then right from that same ganglia, we have output to an effector, and we get a response to our stimulus. The beauty of a short arc reflex like this is it's very fast. We don't have to go mucking about in the central nervous system. We don't have to go through 37 synapses in the brain to make this happen. So we get a very fast reflex. Now, downside is the autonomic ganglion doesn't know about very much, and it can't do much processing. So even though these short reflexes are fast, they're kind of dumb, right? They don't involve a lot of information. But they have a tendency to keep things stable, right? It's kind of like an autopilot. Now, the other kind, the long reflexes, do involve the central nervous system, either the spinal cord or the brain. And here, receptors in the peripheral tissue send signals into the CNS. We get information processing in the CNS, and then from the CNS we go out to the ganglia, and then from the ganglia to the effector. Benefit, we can have complex reflexes that involve large parts of the body, right? Because the CNS is connected to everything, okay? Downside is long reflexes they take longer. In time, they are slower, okay? So the responses happen over seconds to minutes instead of milliseconds to seconds like we see in the short reflexes. Okay, so the long reflexes typically involve more area of the body, multiple organs, or multiple tissues. Okay, so they're encompassing a broader view of input and output. Okay, so if we have these reflexes controlling things, here's some examples of the reflexes, right? So some of the things your autonomic nervous system does. Cardioacceleratory reflex we're going to talk about quite a bit in our cardiac chapter. Pupillary reflex we're going to talk about, you're going to check a lot for in your clinical lives, right? This is the reflex between light entering and touching the retina and size of the iris, right? Size of the pupil. The sexual function is, of course, viscerally reflexive. It includes both short and long reflexes. And then we have, like in our parasympathetic, we have defecation, urination. We're going to talk about many of these reflexes as we go. I put them up here more so that you can see that there are some examples of these autonomic reflexes. Okay, so if we have stimuli that have information processing and then create an output or a response, well, we need to have a way to detect change. How do we detect the stimuli in the first place? So the autonomic nervous system has a number of types of receptors, but they fall into a few big classes. The first big class is the baroreceptor. Baro as in pressure, like barometer, right? A barometer measures pressure, or bariatric surgery is surgery ultimately about reducing pressure from obesity. So baroreceptors detect pressure change and send that information into the central nervous system, autonomic nervous system, or ganglia. All right, so where do we have these? The ones we're going to talk about the most are the baroreceptors in the sinuses, the carotid sinus and the aortic sinus. These are not the sinuses in your head, okay? These are, we just use that word because they're kind of opening. But carotid sinus and aortic sinus detect blood pressure, all right? And we'll talk at length about why blood pressure is important another day. But this is where the detection of that blood pressure is going to come from. We have baroreceptors in the lungs. They are there to provide information about our breathing effectiveness, okay? Again, a topic we will talk about at length is how pressure variance drives airflow in the lungs. So if that's true, we need a way to detect pressure, so we have baroreceptors in the lungs. We have them in the colon and in the digestive tract, and they provide material or information about how much material is present in a particular area of the GI tract, right? So, for example, in the defecation reflex, there are baroreceptors in the rectum that detect when the rectum is full, right? When it's been filled by the colon. The bladder wall, right? We're all familiar with those baroreceptors, right? They detect the pressure of urine in the bladder. The bladder has an amazing ability to relax and expand, but at some point it can't expand anymore and the pressure starts to increase. That pressure increase is detected by the baroreceptors, which send their signal into the spinal cord to prepare for the urination reflex. Okay, so these are the baroreceptors, pressure detectors. The other kind of receptor that plays a major role here are the chemoreceptors. Not surprisingly, they detect chemical changes, okay? The ones we speak of the most, again, carotid bodies and aortic bodies. These areas scan the blood for pH, PCO2, and PO2. In other words, how many hydrogen ions, how much CO2, and how much oxygen is present in the blood that's circulating. You can imagine that during intense exercise, let's say, your hydrogen ion count is going to go up, your CO2 is going to go up, and your oxygen is going to go down, right, because of cellular metabolism. These chemoreceptors detect that, send a signal to the brainstem. The brainstem responds by increasing your respiratory rate and depth, increasing your heart rate, and increasing your blood pressure, right? All to provide better delivery of oxygen and better removal of CO2, okay? So chemoreceptors play a major role in the respiratory and cardiovascular systems and keeping everything in balance. Because it would be silly for us to breathe as fast as we can all the time, right? That would be a waste of energy. But we need to breathe as fast as we can when we're at maximal exercise, right? So how do we detect that? Chemoreceptors. Another place we have chemoreceptors is in the brain, the respiratory centers in the medulla oblongata, which we'll talk much more about later on. They too have the direct ability to measure pH and CO2, to such an extent that any change in pH, regardless of the cause, will result in a change in respiration. So the classic example of this is what we call diabetic ketoacidosis, right? So in that condition, patient has very high blood sugar, but their cells are starving, okay? So they become very acidotic. Their pH is very low. Their number of hydrogen ions is very high. Well, the brain senses that, and even though there's no respiratory issue, the response is to breathe faster and deeper. So a patient who has very high blood sugars and ketoacidosis will be huffing and puffing like they have a pneumonia or a respiratory problem, right? Again, much more on that as we go. All of these cells kind of work in the same way, and that's the cells are affected by their local environment. So if the local environment has a low pH, they respond with stimulation, at least until they get too sick from the lots of hydrogen ions to function properly, right? Which absolutely happens in the brain. Okay, so baroreceptors and chemoreceptors supply the autonomic nervous system with its key bits of information. Okay, so who's doing the decision making then? Well, I would say that the center of the autonomic nervous system is the hypothalamus. Okay, the hypothalamus ends up getting most of the information the autonomic nervous system brings in. The hypothalamus is also heavily connected to every other part of the brain. All right, we're going to hear a lot about the hypothalamus in this class because it pretty much runs our unconscious show. Right? It controls our hormones, it controls our blood pressure, our body temperature, hunger, thirst, all that good stuff is in the hypothalamus. Now, the hypothalamus, though, doesn't have direct connection to the autonomic nervous system. It has to pass its decision-making down through the pons and the medulla oblongata into the spinal cord and then finally out into the autonomic nervous system. Now, as I said earlier and yesterday as well, the hypothalamus by itself cannot know what is going on around you in terms of what are the stressors, what are the potential problems. So the cerebral cortex connects with the hypothalamus. Of course, that's your higher order functions, okay? The limbic system, which is our emotions, memory. The way I like to think of the limbic system is it's our animal brain, right? It's the brain that does all the same things that our friends the animals do. It has profound effects on the hypothalamus. So you can imagine, like in our leopard jumping out of a tree, for long before we were human, that would have responded in a fight or flight scenario, right? So it's a very old part of the brain, but it works very well. The thalamus, its role, if you remember, is to relay information and selectively ignore that which doesn't matter, right? So the thalamus connects with the hypothalamus and all the other sections as well. So this system is complex. You'll find that as soon as you get into the central nervous system, things get very, very complicated, right? Everything is involved and there are lots of interconnections. This is no different. But cortex, limbic system, thalamus, hypothalamus, brainstem, all playing a role in the control of the autonomic nervous system. All right. So in my last 10 minutes, I like to, in this particular talk, give you kind of a preview. You know, I'll say to you multiple times during this class that this isn't clinical medicine, right? My job is not to teach you how to take care of people. That will happen later. But the autonomic nervous system is so important because, well, okay, let me rephrase it. The autonomic nervous system is very important to the body, and because of that, we have developed medicines to affect it in order to accomplish particular goals, right? So just some examples, lots of text on here, but I'll go through it with you. Albuterol. Albuterol is the rescue medicine for asthmatics, okay? Somebody who's having an asthma attack, they're having trouble breathing, they're wheezing, they're having trouble getting air out. What is the problem? Well, the problem is the tubes that carry air into the lungs have been constricted by muscular activity, by smooth muscle. Now, why is that? Usually it's a mistake, right? The body is treating something that is innocuous, like ragweed pollen, like it's some terrible invader that's going to attack you and make you die, okay? So, beta-2 adrenergic agonists like albuterol, they stimulate the beta-2 receptor, but they don't stimulate the other receptor types, the other adrenergic receptors. So we get only beta-2 effects, and because we give it typically in an inhaled way, we only have effects at the lungs, because that's the only place the medicine is, right? So we activate beta-2 receptors just in the lungs. That causes inhibition of bronchoconstriction. How? It reduces the amount of calcium that's present in that smooth muscle cell. Therefore, it's more relaxed. Therefore, that tube is bigger, right? Allowing more air to flow through and relieving the symptoms of poor airflow. So the other place we use this is in emphysema, right? Now, the pathophys is a little different, but it accomplishes the same thing. Beta-2 receptors cause those airways to get bigger, more air can flow and flow more easily. Okay, so that's one example. Another is the beta blockers. I hinted about this the other day. One of the most commonly prescribed medicines in the world. And the reason for that is because a number of big studies have been done which have shown that being on a beta blocker, if you're a person who has had, let's say, a heart attack, dramatically reduces your risk of dying, right? So, okay, there's good evidence. There's something maybe to do. So we have a lot of patients who are on beta blockers. How do beta blockers work? They block beta-1 receptors, most strongly anyway. And by blocking the beta-1 receptors, it reduces the amount of work that the heart has to do to push blood around the body. This is good, particularly if you have a weak heart because you've had a myocardial infarction, let's say. So beta blockers work directly on the beta-1 receptor. Now, interestingly, none of our receptor agonists or antagonists are entirely specific. So like albuterol, we call it a beta-2 adrenergic agonist. If you give somebody enough albuterol, their heart rate will go up. If you give them a lot of albuterol, it'll go up substantially. Why? Because there's some bleed over effect. Some beta-1 receptors are activated by albuterol. Well, beta blockers, same problem. Some beta-2 receptors are blocked by some beta blockers. Okay, so why is that a problem in asthmatics? Well, if you've got a patient who has asthma, if they're not having an asthma attack, no problem. But let's say they're on a beta blocker because they had an MI. Then the ragweed season picks up. They start to wheeze. You give them albuterol, only the beta-2 receptors are blocked by your beta blocker. See the problem? So now you don't have any way to relieve their bronchoconstriction. Now, we've gotten better with our beta blockers. They're much more specific to beta-1, but it's still a relative contraindication. If a patient is an asthmatic, we try not to put them on a beta blocker for this reason. Okay, another example, one maybe you haven't heard of, alpha blockers. Trazocin is an example of this. This is an alpha-1 receptor blocker. Okay, I said alpha-1 receptors are stimulatory, found primarily on in the smooth muscle of the arteries, right? So we constrict arteries, blood pressure goes up. Well, if we block the constriction of arteries, blood pressure goes down, right? So alpha-1 blockers reduce blood pressure. Now, we don't use them very much for blood pressure control anymore because they cause a different problem, which is called orthostatic hypertension or hypotension. If your alpha blockers are all blockaded and you go from lying to standing, your blood pressure falls and then you faint, right? So that's not the greatest thing. But we have found other uses for alpha-1 receptor blockers. So for example, BPH is benign prostatic hypertrophy. It's something that will affect most men as they get to become elderly. This is where the prostate enlarges, and as it enlarges, it starts to impinge on the urethra, which passes through it. Okay, so what the heck does a blood pressure medicine have to do with that? There are a lot of alpha receptors in the prostate. So if we put somebody on trazodone, that reduces the pressure inside the prostate and makes it easier for urine to pass through, therefore relieving some of the symptoms of BPH, right? So there's another example of autonomic nervous system effect in medicine. Okay, another more complicated one, our friend Sudafed, right? There's a medicine with a history, right? It used to be that you didn't have to sign 3 documents and produce proof of United States citizenship to get Sudafed. Well, you do now, right? Why? Of course, that's methamphetamine, right? Can be made from Sudafed. But how's it supposed to work? Well, here, instead of being a direct receptor effector, Sudafed causes an increase in the release of norepinephrine in sympathetic nervous system postganglionic neurons. So if you stop and think about that, okay, more release of norepi will cause more effect at the sympathetic nervous system effectors, right? And it does. Now, what are we trying to do with our Sudafed? We're trying to cause a little vasoconstriction in the mucosa, right? If we restrict the vessels in the mucosa, we get less fluid leak. Less fluid leak is less mucus and secretion development, right? That's our goal. But what we end up with, and that does work, but we also can get anyway, an increase in blood pressure, right? Which is why if you have a patient who has high blood pressure anyway, you don't give them Sudafed if you can avoid it, right? Because while it will reduce nasal congestion through vasoconstriction, it will increase their blood pressure through vasoconstriction. See? So same effect, 2 different places, 2 different problems. Okay, so that's pseudoephedrine, Sudafed. Epinephrine is probably the most important resuscitation drug there is. In the new algorithms, which you will learn in December for how to bring a person back to life-sustaining rhythm, right? You're going to give epinephrine every 3 to 5 minutes until the patient has a blood pressure again, right? So the mechanism of action of epi is clear. It activates all the sympathetic nervous system receptors, and it activates them very strongly. Okay, so it's a way to increase the stimulation of the heart as well as the vessels. All right, a couple of more. Alpha-1 agonists. Okay, this is phenylephrine, also known to its friends as Afrin, right? So this is the nasal spray, okay? And it works similar to the way Sudafed works. If you spray a bunch of alpha-1 agonists up into the vessels of your nose, well, what will happen? They will constrict, right? Constricted vessels make less mucus. Therefore, your mucus production goes down. And we get, nasal clearing. Now, the problem with this system is cells adapt to it, right? So if you use Afrin day after day after day after day, right, your cells, they stay constricted, but they're also adapting to the presence of this blocker all the time. Okay, so then you stop taking the Afrin because, you ran out or you think your cold is done. And then what happens? Well, all those vessels, they dilate again and flood your nose with mucus. So we call that rebound congestion. And it's one of the ways people can actually get hooked on Afrin, okay? Because when they stop it, they get extra congestion more than they had to begin with. So the lesson there is if you're going to use a decongestant nasal spray, use it for an incredibly short amount of time, right? Never for more than a day or 2. And then finally, another place we see autonomic nervous system effectors is in ophthalmology. Many of you who wear glasses or contacts like I do have had your eyes dilated. Well, how the heck did they do that? They use an alpha-1 agonist, and that causes activation of the alpha-1 receptors, which causes your pupil to dilate. There are times when we want to constrict the pupil, like if we're treating glaucoma. In this case, we use acetylcholine. We use the parasympathetic nervous system effector, and by putting acetylcholine directly in the eye, we get pupillary constriction. Now, the downside with that is it doesn't last very long. Acetylcholinesterase is very prevalent in the body. So if we want to have a pupil be constricted for a long period of time, we have to kind of doctor up the acetylcholine so it's resistant to the acetylcholinesterase blockers. Okay?